Hello, everyone, and welcome to today's live broadcast, Laboratory Diagnosis of von Willebrand Disease, presented by Dr. Larry Smith, Manager, Medical and Scientific Affairs, Hematology, Abbott Diagnostics. I'm Alexis Corrales, and I will be your moderator for this educational web seminar presented by Labroots and sponsored by Abbott Diagnostics. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button in the bottom left corner and follow the process to obtain your credits. I'd like to now introduce today's speaker, Dr. Larry Smith. Dr. Larry Smith received his doctorate in biological sciences from Fordham University, Bronx, New York, and completed his certificate in molecular laboratory diagnostics at Michigan State University. He completed his postdoctoral studies in gene therapy and experimental hematopoiesis at Columbia University College of Physicians and Surgeons, New York, and was a research fellow in immunology at Fordham University. In addition, Dr. Smith has a Master of Science in Medical Biology and a Bachelor of Science in French and Biology. He holds multiple board certificates and has published numerous peer-reviewed articles, abstracts, reviews, and monographs. His research interests include laboratory test development for hemostasis and coagulation, HPC therapeutic applications, and flow cytometry. For a complete biography on Dr. Smith, please click his photo in the top left corner of your screen. Welcome, Dr. Smith. You may now begin your presentation. Please join me in welcoming Dr. Larry Smith. I will now turn the presentation over to him.
Alexis, thank you very much for that introduction, and I'd like to thank all of the audience for attending today's lecture. This is on laboratory diagnosis of von Willebrand disease. These are my disclosures. Actually, I have nothing to disclose. I do lecture for ASCP, and I'm currently an adjunct professor at Rutgers University in New Jersey. Now, prior to joining Abbott, I was director of the Coagulation Laboratory, assistant director of Hematology and Flow Cytometry at Memorial Sloan Kettering Cancer Center. What I'd like to do over the next 45 minutes is to define von Willebrand disease and tell you a little bit about its history. We'll review the current classification of von Willebrand disease and discuss some of the assays that are utilized in the diagnosis of this disorder. We will review the structure and function of the protein, and then we'll look at some of the methods that are very essential in the diagnosis of von Willebrand disease. We'll look at some of the limitations of those methods. We'll look at some pre-analytical variables that influence the testing in the laboratory, as well as how we interpret the results from those assays. So to start with, von Willebrand disease is a disorder of primary hemostasis. We know that we have disorders of primary hemostasis that involves blood vessels and platelets, and we have disorders of secondary hemostasis that involve the coagulation factors. So here we're just going to focus on those disorders in the primary hemostasis realm. Von Willebrand disease is the most common congenital bleeding abnormality in the world. In terms of frequency, it affects about 1 to 2% of the general population. But not everyone who has von Willebrand disease will actually bleed unless they're challenged. It's only symptomatic in about 1 in 10,000 individuals. In terms of its history, it was described by Dr. Eric von Willebrand, who was an internist working in Finland, and he was taking care of a 5-year-old girl from the Aeolian Islands who presented with a bleeding disorder. Now, this bleeding disorder appeared to have somewhat of an autosomal dominant pattern of inheritance. She presented with mucocutaneous bleeding, a prolonged bleeding time, and a family history of bleeding. Now, unfortunately, she died in her early teens from increased menstrual bleeding. Dr. von Willebrand, however, did recognize that this bleeding pattern that this little girl presented with was a little different from the classic hemophilia that they had diagnosed in the past, and usually boys presented with this type of bleeding disorder. And so with that, he decided that she had what appeared to be an autosomal mode of inheritance, and he actually wrote a paper about it in 1926, and he wasn't really sure what to call it, so he called it pseudo-hemophilia. Now, as a disorder of primary hemostasis, some of the clinical manifestations will include mucocutaneous bleeding that's of varying severity in both males and females. Some of these characteristics will be ecchymosis, epistaxis, gastrointestinal bleeding, gingival bleeding, and menorrhagia in females. The basic pathophysiology that we see in von Willebrand disease is due to a defective platelet adhesion and a concurrent reduced level of factor VIII. This slide shows you the actual structure of the von Willebrand factor gene that's responsible for this protein. The protein itself is a large multimeric protein, and it ranges in size from 600 Daltons all the way up to greater than 20 million Daltons. So it's a really large protein that's produced. It's produced on the short arm of chromosome 12, and it consists of about 178 kilobases with 52 exons. Now, if you look at the middle of the screen, we can actually see the structure of the gene that's responsible for von Willebrand disease. And as I said again, it's synthesized on chromosome number 12, the short arm. There's also another gene that can give rise to von Willebrand factor, and this is a pseudogene that's located on chromosome 22. The next diagram in the very middle of the screen represents the complementary DNA. The DNA was cloned, the gene was actually cloned in the late 80s or so, and this represents the structure of that complementary DNA. As you can see here, it consists of two regions, a polypeptide region and a mature subunit. These sections are made up of various domains, D1, D2, D prime, D3, etc. And this is a very heterogeneous disorder, so mutations can occur along any of these particular domains. We can also see where the von Willebrand factor interacts with factor VIII, that's somewhere here within the D prime and part of the D3 domain. We can see where it interacts with the glycoprotein 1b receptor of the platelets in this particular region, and also with the glycoprotein 2b3a receptors of platelets in this particular region here. In addition, von Willebrand factor interacts with collagen in two different regions that we can see at the very bottom of the slide. And now understanding where these mutations occur is very essential because it results in different phenotypic expressions of von Willebrand disease. A mutation that occurs here in the D prime region would result in a presentation that we call type 2N von Willebrand disease, where there is an abnormal interaction with the factor VIII molecule. A mutation that occurs in this A2 domain would result in something that we call 2A von Willebrand disease. So these really help us in terms of actually diagnosing these patients when we understand where these mutations occur. In terms of the synthesis of von Willebrand factor is synthesized in two different regions. It's synthesized in endothelial cells. We know that endothelial cells make up or surround the actual blood vessels in our bodies, and it's also synthesized in megakaryocytes. Now, once it's synthesized, it's actually stored in an organelle within the endothelial cells called the Weibel-Palade bodies. In megakaryocytes and platelets, it's stored in the alpha granules. Now, as the protein is synthesized, it starts out as a monomer or single chain. This is called the pre-pro von Willebrand factor monomer. As it begins to move to the endoplasmic reticulum, that is, move out of the nucleus into the endoplasmic reticulum, the signal peptide region is cleaved off, and then this monomer actually undergoes a dimerization. So we go from one chain to two chains. These two chains are then are linked together at their carboxy terminal end. And then as the protein then leaves the endoplasmic reticulum and moves on to the Golgi apparatus, it undergoes a multimerization. So that just simply means that we go from two chains or a dimer into 4 chains, 8, 16, 32, 64, etc., until we end up with this really large molecule. Now, once it's fully synthesized, the propeptide region gets cleaved off, and then this large structure becomes very tightly coiled, and then it's stored in the Weibel-Palade body. Now, in terms of the release of von Willebrand factor, there are a number of theories as to how it actually gets out of the Weibel-Palade body into the circulation. And so it really doesn't matter which one of the theories you believe, but if you look at the area where I'm pointing to, that section D on the slide here, there is one theory that the Weibel-Palade body moves up to the region where the damaged endothelium has occurred, and then the Weibel-Palade body releases these very long strands of von Willebrand factor. Now, these very long strands, or these unusually large multimers of von Willebrand factor, can actually be very dangerous because they're very adhesive to platelets. So in order to prevent platelet agglutination from occurring within our blood vessels, located on top of the endothelial cell, and this is the figure on the right side of the slide, is a protein that's called ADAM-TS13. It's also called the von Willebrand factor cleaving protease. And as it appears on this slide, it looks like a pair of scissors in a sense. And so as these unusually large multimers of von Willebrand factor are released, the pair of scissors, or the ADAM-TS13, sits there and cleaves this protein into much smaller fragments. And so the smaller fragments of von Willebrand factor then are not as adhesive to those platelets, and so then we have the normal von Willebrand factor in our circulation. Now, in terms of the function of von Willebrand factor, there are actually only 2 functions that have been assigned to von Willebrand factor. The first one is that it serves as a ligand that binds to the glycoprotein 1b receptor of platelets. Once it binds to those platelets, it will then pull the platelet down to the damaged endothelial layer. So it actually binds to the glycoprotein 1b receptor as well as to the collagen that's now exposed in the damaged endothelial layer. This particular function leads to what's called platelet adhesion, where we form a monolayer of platelets on the damaged endothelial surface. The second function of von Willebrand factor is that it serves as a carrier protein to factor VIII. So, in other words, it protects factor VIII in the circulation. Factor VIII, once it's produced, will come out into the circulation, and von Willebrand factor will then simply wrap around it to protect it from unnecessary cleavage or proteolysis. And then it also co-localizes this factor VIII to the site of injury so that factor VIII can then function in the coagulation cascade. Now, moving on to some of the assays that we use to classify von Willebrand disease, I've actually divided these assays into 3 groups. These are all screening assays, factor VIII C, von Willebrand factor antigen, and von Willebrand factor activity. These are all necessary assays for the most basic diagnosis of von Willebrand disease, and I'm sure all of your laboratories do all 3 of these assays. In the box in the middle, I have some additional assays that can be used in the diagnosis of von Willebrand disease, so they are just an adjunct to the first 3 assays. This would include the bleeding time and or a platelet function screen, or PFA 100, PFA 200. I would imagine that most of us no longer do an actual bleeding time, and so we've all switched to the PFA 100/200 to perform or to replace the bleeding time. And in the third box that's on the right, there are two other assays that are listed here. Multiple analysis. This is somewhat a little more complicated than the other two boxes, and certainly platelet aggregation. These also will aid in further classifying the type of von Willebrand disease that we're talking about. Understanding these assays then and performing these assays will then allow us to classify von Willebrand disease. So we classify von Willebrand disease into two broad categories. We have the disorders that appear to be a quantitative defect. So a quantitative defect would be a decrease in the amount of protein that's present, but the protein is normal in terms of its function. Now, under this category, we have something called type 1 von Willebrand disease, and this is a partial quantitative deficiency of the disorder. And then we have type 3 von Willebrand disease. This is a complete absence or very severely decreased amount of the von Willebrand factor protein. The second broad category would be those disorders that are due to a qualitative defect. So here we have a defective protein or dysproteinemia. Now, we may have a normal amount of the protein, or it may be decreased somewhat, but the main point is the protein itself is defective. Now, these are divided into four subgroups. We have type 2A, type 2B, type 2M, and type 2N. So let's look a little further into these actual categories of von Willebrand disease. So we'll first look at the quantitative defect. So again here, we have a normal protein in terms of its function, but we have a decreased amount of that protein. So the first disorder that we'll look at is called type 1 von Willebrand disease. This is the most common presentation for patients with von Willebrand disease. About 80% of patients who do present with the disorder will fall into this category. It's characterized by a heterozygous mutation that leads to this partial quantitative deficiency. So if you can imagine, we have two alleles that are present, and if one of the alleles is, and each one of these alleles will produce, let's say, about 50% of the von Willebrand factor that we have. So if one of those alleles is mutated or one of those alleles is absent, then the remaining normal allele will produce its 50% of von Willebrand factor. So as a result, we have a reduced amount. Since von Willebrand factor protects factor VIII in the circulation, we'll also have a concomitant decrease in the amount of factor VIII because we just don't make enough of the von Willebrand factor to protect it in the circulation. Now, in terms of how these patients present in the clinic with type 1 von Willebrand disease, their bleeding symptoms can range from asymptomatic, in other words, we don't see any bleeding at all unless they're really challenged, up to just a very mild bleeding diathesis. So a very wide range in terms of bleeding symptoms when they present to the clinic. Now, understanding that this is type 1 von Willebrand disease is also very advantageous because it's very easy to treat these individuals. Since the protein that's produced is a normal protein, but just not enough of the protein, we can simply give these individuals a drug called DDAVP, diamino-8-dearginine vasopressin, which is just simply, we can give it in the form of a nasal spray. They spray it into their nose, and this then will cause the endothelial cells to release more of the von Willebrand factor from those endothelial cells into the circulation. It's okay because this is a normal von Willebrand factor protein in terms of its function, so a real easy solution for treating patients with type 1 von Willebrand disease. What would we see in the laboratory in a patient that presents with type 1 von Willebrand disease? Well, again, we only have a slight reduction, so we could expect to see a normal to a decreased level of factor VIII, a normal to decreased level of von Willebrand factor activity, or if you call that the ristocetin cofactor assay, that would be normal to decrease. And we'd also see a normal to decreased amount of the actual protein that's measured by the von Willebrand factor antigen assay. Now, I say normal because it all depends on what your lower limit is on your reference range. If your lower limit is 50% and you have one allele that's producing von Willebrand factor, then there's your 50%. If that one allele produces 40% or 35%, then that's what you would have, and then that level might be considered decreased. But the range is normal to decrease for these three tests. If you were to do a bleeding time, usually in type 1 von Willebrand disease, the bleeding time is prolonged. And so that would translate into a PFA 100 also having a prolonged closure time. And then finally, if you were to do multiple analysis, if you look at the cartoon on the right side of the screen, when you do multiple analysis, and I'll explain it in a little more detail on a later slide, but we're simply looking at we're placing a sample on a gel, applying a circuit, and we're separating the multimers of von Willebrand factor based on their size. And so what you can see here in this diagram is in lane 1 that says normal with the blue dots around it, we can see that we have 3 sizes of these von Willebrand factor multimers. And so where I'm circling at the top would be the large molecular weight multimers, and then we have the intermediate molecular weight multimers just below, and then below that, the really large multimers at the bottom would be the low molecular weight multimers. So high molecular weight, intermediate molecular weight, and low molecular weight multimers. All 3 sizes of these multimers are present. When we look at someone with type 1 von Willebrand disease, where we see the red box in lane 2, we see that we still have all 3 sizes of these multimers present. The high molecular weight multimers, the intermediate, and the low molecular weight multimers. But note, in terms of the intensity of the staining of each of these individual bands, it's a lot less. So therefore, in type 1 von Willebrand disease, if you were to do multimeric analysis, what you would find is that all 3 sizes of the multimers are present, but you just don't have as much of the protein when you compare it to someone that's normal. Okay. Now, moving on to type 3 von Willebrand disease. This is the most severe form of von Willebrand disease, and fortunately, it only affects about 1 to 5% of the general population. And it's usually seen in cases where there's an increase in consanguineous marriages, so that's where it's more prevalent. Now, this results from a homozygous mutation that leads to a complete deficiency of von Willebrand factor. So now if you can imagine 2 alleles, and remember that each allele will produce about 50% of whatever amount of von Willebrand factor that you have, if one of the alleles is missing, or both are missing, or if one is missing and one is defective, or if both of the alleles are defective, you can see then that you would not be able to really produce any von Willebrand factor. When we measure the von Willebrand factor levels in these patients, it certainly ranges from 0 up to somewhere less than 5%. Now, since factor VIII is protected by von Willebrand factor in the circulation, and now we don't have any von Willebrand factor, then you would expect to see a much greater decrease in the level of circulating factor VIII. And usually these levels are low, but they're not as low as what we might see in someone with severe hemophilia. Since von Willebrand factor then also plays a role in platelet function, and if we did not have any of this von Willebrand factor, you would expect that these patients might present with some spontaneous bleeding, certainly severe mucocutaneous bleeding, since this is one of the characteristics of primary hemostasis. And then these patients also can bleed into the soft tissues as well as into the muscles and into the joints. So, and this particular bleeding pattern might be consistent with hemophilia, but again, this would not be hemophilia since it's type 3 von Willebrand disease. Now, turning to the qualitative effects, these are a little more complicated than the quantitative effects. So here we have an abnormal protein that's present. So there's something that's intrinsically wrong with the protein itself, that it doesn't function properly. However, the levels of this protein may be normal to decrease. Very often they're normal levels. So the first disorder that we'll look at is called type 2A von Willebrand disease. Now, type 2A von Willebrand disease results from a mutation that occurs in the A2 domain. You remember when I showed you the cDNA and we saw all of the domains? So this mutation would occur in this specific region. And this is a region where the von Willebrand factor interacts with platelets as well as with collagen. So here we have a loss of the platelet-dependent function. If we were to look at multimers in these patients, what we would see is that when we look for the high molecular weight multimers, they are missing. When we look for the intermediate molecular weight multimers, they are missing. And we only see the low molecular weight multimers in this scenario. Now, there are several theories as to what happens to this von Willebrand factor or to these multimers. And one of the theories is that there is an abnormal assembly and secretion of the protein. And a second theory is that once this protein is produced, there's an increased susceptibility of this protein to proteolytic cleavage once it gets into the circulation. Now, since we have lost those higher molecular weight multimers, which have this platelet adhesive quality, you would expect to see an increase or somewhere from a moderate to severe mucocutaneous bleeding in these patients. The next disorder would be type 2 von Willebrand disease. Now, here the mutation occurs in the 81 domain of the von Willebrand factor. Now, this is a very tricky mutation because it results in a loss of only the high molecular weight multimers. Remember, in type 2, we lost the high and the intermediate, but in type 2A, but in type 2B, we only lose the high molecular weight multimers. Now, this mutation that occurs is called a gain of function mutation. Now, so that means that the mutation that's occurring in the von Willebrand factor is such that it has an increased affinity to bind to the glycoprotein 1B receptor of the platelets that are just simply floating by or circulating in the blood. So the von Willebrand factor jumps to grab onto the platelet. This results in spontaneous binding of these platelets that are taken out of the circulation, resulting in thrombocytopenia, and in addition, there's this rapid loss of those large von Willebrand factor multimers. Now, as you can see, this particular type of von Willebrand factor is really abnormal. So in terms of treating these patients with type 2 von Willebrand disease, we would definitely not want to give them DDAVP because we don't want to put more of that von Willebrand factor into the circulation. So DDAVP would be contraindicated in type 2B von Willebrand disease. The next type is called type 2M von Willebrand disease, and here there's a mutation in the A1 binding domain, and this simply leads to a decrease in platelet function. Now, I tried to schematically draw what this would look like, and as you can see it on the diagram on the right, the mutation occurs and prevents the glycoprotein 1B from binding to the von Willebrand factor. But note that where von Willebrand factor binds to factor 8, there is no abnormality there. So if we were to test for the level of factor 8 in these patients, if we were to test for factor 8 in these patients, the factor 8 would be normal. If we do multimeric analysis, we would have high, intermediate, and low molecular weight multimers present in these patients, in this particular category. So we just simply have a defect in the platelet-dependent function in this scenario. The next type is called type 2N von Willebrand disease. This is also referred to as autosomal hemophilia or the Normandy variant. It was actually described in Normandy in France. Now, the mutation here is where von Willebrand factor binds to factor VIII. If you look to the right side of the cartoon there, you see I put a little arrow here to show you that factor VIII cannot bind to the von Willebrand factor. But note that in terms of platelets binding to the von Willebrand factor, there is absolutely no problem there. So there's normal platelet adhesion, but the von Willebrand factor simply cannot protect factor VIII in the circulation. So now if we go back to the laboratory and test patients with type 2N von Willebrand disease, as we would suspect, we'd have a decrease in the level of factor VIII. And this would be similar to like a mild hemophilia. If we measure the amount of von Willebrand factor antigen and the von Willebrand factor activity, these would be normal because we have a normal amount of the protein, but it's just a dysproteinemia that we're looking at. And if we do a bleeding time, or PFA 100, in patients with type 2N von Willebrand disease, we actually get a normal result. So this would be one of the only categories of von Willebrand disease where the bleeding time is really normal. And that's because there is no problem with platelet adhesion in these patients. Now, the next disorder that we'll talk about very quickly is called Pseudo-von Willebrand disease, or platelet-type von Willebrand disease. And this is not really a defect in von Willebrand factor itself, but rather a defect within the platelets. So here, von Willebrand factor is normal. Now, the defect or the mutation occurs in the platelet glycoprotein 1b receptor that results in a gain-of-function mutation. So if you remember the gain-of-function mutation that we talked about with type 2B von Willebrand disease, well, this same type of mutation occurs here in the platelets. But this time, the platelets have an increased binding affinity for the von Willebrand factor. So the von Willebrand factor is normal, and the platelets jump to grab onto the von Willebrand factor. So the presentation is identical to what we see with type 2B von Willebrand disease. But in this case, this is not von Willebrand disease. In terms of the laboratory findings, they're all the same. Since the platelets are going to bind to the von Willebrand factor, we're going to lose the high molecular weight multimers when we look at multiple analysis. We're going to have thrombocytopenia because we're pulling the platelets out of the circulation. And a test that I have not mentioned, but I will talk about it a little later, is platelet aggregation using low-dose ristocetin. This particular assay really does help to identify patients with pseudo-von Willebrand disease as well as type 2B von Willebrand disease. So here's a case where platelet aggregation using various doses of ristocetin, but in particular a low dose of ristocetin, would help us to differentiate these two presentations of von Willebrand disease from all of the others. Now, since the defect here is in the platelets, we would not want to give these patients the same treatment that we give for patients with true von Willebrand disease because we'd just simply be adding fuel to somewhat of a fire if we did so. Now, at the beginning of the talk, we said that von Willebrand disease was the most common congenital bleeding abnormality in the world. But there is an entity that's called acquired von Willebrand disease. So any of you that are listening to this can actually acquire von Willebrand disease. And it results in a qualitative structural functional defect within the von Willebrand factor molecule, and it leads to an increased risk for bleeding. There are several mechanisms to explain how this can occur. And the first one is due to an autoimmune clearance, an antibody that forms that inhibits the von Willebrand factor and leads to an increased clearance of the von Willebrand factor from the circulation. Some of the disorders where this is seen would be some of the lymphoproliferative disorders, such as chronic lymphocytic leukemia, MGUS, the monoclonal gammopathy of unknown specificity, and certainly patients with systemic lupus erythematosus. Another mechanism that can lead to acquired von Willebrand disease is a low-functioning thyroid gland, so hypothyroidism. Now, this is a non-immune-mediated mechanism, but the result would be the same. Often when patients have aortic valvular stenosis or they have a left ventricular assist device, these result sometimes in a fluid shear stress induced proteolysis, whereby the valves are kind of hard and they clamp down on the von Willebrand factor, causing it to be uncoiled, leading to its cleavage. So this would deplete some of the larger molecular weight multimers of von Willebrand factor. Two other mechanisms that are associated with acquired von Willebrand factor would be an increased binding of von Willebrand factor to the surfaces of platelets. So in one of the myeloproliferative neoplasms, essential thrombocythemia, we have platelet counts that can get up to over 1 million, and just having that surface there can draw some of that von Willebrand factor out of the circulation. And then finally, there are a number of drugs that have been associated with an acquired von Willebrand disease. Now, one of the things that you'll look for in the laboratory certainly is the ratio of the activity to the antigen does not always match. So we have this discrepant ratio. And so this is really very, very characteristically seen in many cases of acquired von Willebrand disease. Now, turning to look at some of those assays that we've already talked about, but to look a little more in depth at the assays, again, we have the screening assays, the bleeding time or platelet function screen, followed by the sensual screening assays, factor VIII, von Willebrand factor antigen assay, the von Willebrand activity assay. Now, this activity assay can be divided into a number of different assays depending on what your laboratory uses. And then we have more conformational assays such as multimer analysis and ristocetin-induced platelet aggregation. And there are others, but we're limited for time, so I just listed some of the more common assays. So just very quickly concerning the PFA 100 or PFA 200, if you look at the diagram on the left, this represents an entered blood vessel. We know that all blood vessels in the body are surrounded by a single layer of endothelial cells, and when we damage that endothelial layer, we expose a layer of subendothelial collagen microfibrils, and we also damage that Weibull-Palade body that stores the von Willebrand factor. As a result, the von Willebrand factor leaves the Weibull-Palade body and comes into the lumen of the blood vessel. It attaches to the subendothelial collagen layer, and then it goes up and looks for those platelets. Platelets bind via that glycoprotein 1b receptor, and the von Willebrand factor is pulled down onto the damaged endothelial surface. It forms this monolayer, and this is the initial protection that occurs. And we know that later platelets will aggregate, and then our coagulation proteins will then come together and form a stable clot here. Well, the same thing kind of happens in the PFA 100, whereby we have a column. We introduce blood into the column, and then there's a little pressure that pushes the blood up through the column. And as these platelets then interact with collagen that's located in the cartridge or with one of the platelet agonists, we know that the platelets will adhere, and then as a result of the collagen, and the platelets will then aggregate as a result of either epinephrine or ADP that's been added to the cartridge. And then at some point, the plug gets to be so large that the analyzer can no longer push the blood up through the cartridge, and that results in a closure time. So it's very, very similar to what happens in vivo. This would be the in vitro, and this would be the in vivo phenomenon. How good is the PFA 100 in diagnosing patients with von Willebrand disease? Well, if you look at all types of von Willebrand disease, with the exception of 2N, we have type 1, 2A, 2B, 2M, and type 3. The PFA 100 has a sensitivity of about 85 to 90%. Now, if we only look at the type 2 and the type 2A, type 2B, type 2M, and type 3, then the sensitivity can go up to somewhere around 98%. based on a number of studies that have been done looking at von Willebrand factor and the PFA100. The PFA100 by itself certainly cannot say that a person has von Willebrand disease, but it will have a consistent activity in the presence of individuals with von Willebrand disease. Turning to some of the other assays, the first assay would be the antigen assay. This assay simply measures the amount of protein that's present. It's an immunoassay, and it generally involves, most of these are latex immunoassays, so they involve latex beads that are coated with an antibody. This antibody is specific for von Willebrand factor. And so when we add a patient's plasma that contains von Willebrand factor, it will bind to these antibodies, and these beads will then ultimately fall to the bottom of the cuvette. Now, all along, our analyzers are shining a light through these cuvettes, and we will be able to measure a change in optical density from that light that's passing through the cuvettes, and then correlate that to a certain percentage of the protein. Now, this von Willebrand factor antigen assay measures abnormal protein as well as normal protein. So it doesn't tell us if the protein is functional, it simply tells us that the protein is present in these individuals. Moving on to the activity assay. The activity assays are based on taking normal platelets, either from a donor or formalin-fixed platelets, adding von Willebrand factor to those platelets, and then the von Willebrand factor will cause those platelets to agglutinate, and then we can look for a change in optical density. The gold standard method for assessing von Willebrand factor activity is the risk-decedent cofactor assay. This assay measures the ability of von Willebrand factor that's in the patient's plasma to induce an agglutination reaction in normal platelets. These again, they could be normal formalin-fixed platelets or normal donor platelets, but these will undergo an agglutination reaction when risk-decedent is then introduced into the milieu. So again, patient's plasma, normal donor plasma, risk-decedent, and then we get a reaction. This is an example of what the agglutination reaction would look like on the platelet aggregometer. So we simply measure the slope of this aggregation of this agglutination reaction that has occurred, and then we correlate that to a calibration curve that we've run prior to running that patient sample on our platelet aggregometer, and this will give us a measure of the functional activity of that von Willebrand factor. Again, this is the gold standard. Now, how good is the risk-decedent cofactor? Well, there are some problems with the risk-decedent cofactor in terms of its coefficient of variation. We know that these can range anywhere from 10 to 30%. But with that being said, it still is the gold standard when it comes to measuring von Willebrand factor activity. Now, a modification somewhat of this assay in order to determine von Willebrand factor activity, there is a latex particle enhanced immunoturbidimetric assay, and this assay uses a specific anti-von Willebrand factor monoclonal antibody that recognizes the region on the von Willebrand factor where the glycoprotein 1b receptor platelets would bind to. And so in this assay, we have latex beads that are coated with that specific antibody. When we add the patient's plasma to the cuvette, if that region is available, it will bind to these latex particles, and then we'll get an agglutination reaction occurring, whereby we'll get an increase in light that's shining through the cuvette, and we can measure that change in the light that's being transmitted through the cuvette. And then this would be read from a calibration curve. Additional assays that mimic the immunoturbidimetric assay, there's one where there is no risk-decedent that's added, and in this particular assay, there's an antibody against the glycoprotein 1b alpha that's captured onto a polystyrene particle. And now this gain of function mutation then would induce the agglutination reaction, and so this tries to do the very same reaction in the absence of ristocetin. And then finally, there's another assay out where ristocetin is added back to the reaction, and it's an immunoturbidimetric assay that uses a recombinant fraction of the glycoprotein 1b alpha receptor. And then the ristocetin is added to induce the binding of this monoclonal antibody to von Willebrand factor that leads to the agglutination, and we can simply measure the change in optical density. The factor VIII assay you all are very familiar with would simply involve taking a patient plasma that has an unknown level of factor VIII, so we get a fairly prolonged clotting time if we were to do an activated partial thromboplastin time on this sample, and then we take a normal plasma that's completely deficient in factor VIII, which would have an extremely long prolonged clotting time, and then we simply look for a correction in the clotting time when we mix these two plasmas together, and then we can correlate the number of seconds that we get to a calibration curve to determine what the level of factor VIII was in the patient's plasma that led to the correction in the mixture. Moving on to multiple analysis, again, this is an electrophoretic assay whereby a sample is applied to an agarose gel. We have different types of agarose gels with different concentration of agarose. We have what's called a low-resolution gel with about less than 1% agarose that leads to a very good separation of those largest von Willebrand factor multimers. And then we can have any combination leading up to a higher resolution gel, which would be very good at separating those triplet structures that we see once we apply an electric charge to this gel. So once the electric charge is applied, then it causes the bands to migrate through the gel, and then we ultimately end up with high molecular weight, intermediate molecular weight, and low molecular weight multimers. These are then fixed using an antibody to von Willebrand factor. They're transferred onto nitrocellulose or nylon or some other material. We then can visualize these, and we can even quantitate the bands using a densitometer if we needed to. One other assay that's a very good assay out there that's used in many parts of the world is the collagen binding assay, and this measures the ability of the large von Willebrand factor multimers to bind to collagen. So it would be a very good assay to kind of differentiate between type 1, 2A, 2B, and type 3 von Willebrand disease. And there's a number of reports that suggest that it's far more sensitive to than the von Willebrand factor ristocetin cofactor assay to the loss of those high molecular weight multimers. And simply it's an ELISA-based assay where the plates are coated with collagen, and then we simply add a patient's plasma and a number of other things, and then we read the amount of light that's transmitted in our ELISA plate reader. A few problems with the assay is that, number one, it's not really standardized throughout. Number two, there are different sources of collagen, so that can lead to some variability in results. The ristocetin-induced platelet aggregation that I mentioned, I can just show you how this really works. It helps us to identify type 2B von Willebrand disease and platelet-type von Willebrand disease from all of the other categories of von Willebrand disease. So in the laboratory, when we do our platelet aggregation, we often use various doses of ristocetin, a high dose and a low dose, the high dose being 1.5 mg per mil and the low dose being 0.5 milligrams per mil. So if you take a normal individual and run 1.5 milligrams per mil, you'll get a very nice deflection of the curve. On that very same normal individual, if you use 0.5 milligrams per mil, you are expected to get no reaction by the flat line that we see right here. So this is perfectly normal. In those patients where there is this gain-of-function mutation, type 2B or pseudo-von Willebrand disease, what you see is that with a high dose of ristocetin, 1.5 mg per mil, we get a very nice deflection of the curve. And when we use 0.5 mg per mil, we also get a very nice deflection of the curve. So by this increase or this hyperactivity to the low dose of ristocetin, this would suggest type 2B von Willebrand disease or platelet type von Willebrand disease. Turning to the pre-analytical variables, certainly phlebotomy conditions are important, patient stress levels. If you were to draw blood from a 5-year-old kid, and the arms are flailing around and the child is screaming and crying, this leads to an increased amount of stress and higher levels of von Willebrand factor in these patients. Certainly, if you draw the blood when they are relaxed and at ease, you'll get a truer reflection of the amount of von Willebrand factor that's in these patients. Sample processing and sample storage are also pre-analytical variables. A study was done by Bowman coworkers to show that the von Willebrand factor antigen, the risdiplam cofactor, and the factor VIII, if they were drawn in blood and left at room temperature for 3 hours, these would be the normal levels. If that sample were placed on ice and then tested in 3 hours, that would appear to be a decrease in the levels. So certainly, you don't want to really place these samples on ice. And we can see the same phenomenon if we look at the multiple studies. We'll look at the one in the middle that says type 1 von Willebrand disease. We see that in the baseline sample, we have high, intermediate, and low molecular weight multimers. When the sample sat at room temperature for 6 hours, again, high, intermediate, and low. But when the sample was placed on ice and then tested in 6 hours, it appears that we've lost the high molecular weight and a real reduction in the intermediate molecular weight, and we're just kind of left with the low molecular weight multimers. So these can artificially lower von Willebrand factor levels if we place them on ice. Certainly race plays a role. Von Willebrand factor levels are higher in African Americans and Africans than in other races. Age plays a role. Von Willebrand factor increases with age, and certainly the ABO blood groups play a role. This was a study done by Gill and co-workers, where they looked at individuals with type O, A, B, and AB blood types. These are the number of individuals that were looked at, ranges from 109 to 456 with each one of the types, and we can see the mean of the normal range that they got from type O being about 75% versus 123 for patients with type AB. And so as a result, if you use their particular range, your cutoff would be about 35.6 for the low end and 157 for the high end. I know that many laboratories generally use something around 50 to 150. So one would just want to consider blood type before saying someone actually has type 1 Willebrand disease, especially if they had type O von Willebrand. They had type O blood. This is just a summary of some of the variables that can affect von Willebrand factor testing. And what you can see here is that it's elevated when it comes to age, chronic inflammation, diabetes, etc., and it's decreased in hypothyroidism and type O blood. So to conclude, von Willebrand disease is a very heterogeneous group of clinical manifestations. It requires a battery of assays, von Willebrand factor activity certainly being probably the most important assay because you want to know whether the amount of protein you have is really functional. The collagen binding assay certainly can contribute, and it's a very important assay. There are a number of supplemental assays that can also be added to the armamentarium for diagnosing von Willebrand disease in the laboratory. So with that, I want to thank you for your attention, and we are ready for any questions that you might have.
Thank you, Dr. Smith, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many of your questions as we have time for. So let's get started. Our first question is multiple analysis needed for all types of VWD diagnosis?
That's a very good question. Multiple analysis is not needed really for every type of diagnosis in von Willebrand disease. If you're looking at someone who has type 1 von Willebrand disease, the analysis is going to come back normal. It would only be if you were to try to determine how much of the protein that's present that's going on with something like densitometry, or actually look at the bands themselves to see that they were somewhat reduced. But multiple analysis is not really needed for all diagnoses in von Willebrand disease, but as you can see from what we've looked at, it certainly helps us to differentiate between type 2A, type 2B. If you suspect that someone has type 3 von Willebrand disease and you do multiple analysis, you're going to get nothing in terms of the multiples. However, many people will order multiple analysis just to complete the picture.
Okay, question number 2. How is acquired VWD diagnosed?
Acquired von Willebrand disease would be diagnosed using the very same assays that we use, but in this scenario, we would certainly look at patient history. If there was no prior history of bleeding, no ecchymoses, no epistaxis, no gingival bleeding, no menorrhagia, etc., and these happen to appear when an individual turns, let's say, 55 or 60 years old, then we could assume that it would be acquired as opposed to being hereditary. Also, if there was no family history, we could certainly use that as a part of the diagnosis of this acquired von Willebrand disease.
What are the different recombinant von Willebrand factors used for treatment?
I can send you an email that explains more of the recombinant factors. This lecture really was based on just diagnosing and not really for treating patients. So if you can send me an email, I can let you know what some of those recombinant factors are and give you the sources where you can find those.
What do you think is the best VWD screening test?
If I were to have only one VWD screening test, that would be one of the activity assays. What's important here is knowing that whatever amount of protein you have, that it functions properly. You can have 100% of a protein that's abnormal, and it doesn't do you any good. So I think one of the activity assays would be the very best if you only had one. But you really need activity, antigen, and factor VIII at a minimum.
So we are almost out of time, so we have time for one more question. Can the APTT assay be used as a substitute for the factor VIII assay?
Absolutely not. The APTT is subject to differences. It's a clot-based assay, so when we have differences in the constitution, let's say the phospholipids or the activator substance, we can get various reactions to different levels of that factor VIII. So one reagent might not prolong above the upper limit of normal until you get down to, let's say, 20% of factor VIII, whereas another one might respond or prolong at 30% or 35%. So it really cannot be used as a substitute for the level of factor VIII.
I would like to once again thank Dr. Smith for his presentation. Dr. Smith, do you have any final comments for our audience today?
It's really been very nice talking to all of you. I hope that you were able to learn something or learn something new, because I'm sure most of you are experts in von Willebrand disease testing anyway, so I hope I was able to bring something to you that you did not know before. We certainly welcome any additional questions that you have, and we'll do our best to provide answers to all of those. So thank you for your participation.
Thank you again for joining us today, Dr. Larry Smith. I would also like to thank Labroots and Abbott Diagnostics for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through June of 2018. You will receive an email from Labroots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Bye-bye.